Hello everyone, and welcome to today's webinar, Socially Distanced Experiments, the importance of single lab accessible automation in driving high quality proteomics research. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Biotechne. To learn more, visit biotechne.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Chris Heger, Director of Applications Science, Analytical Solutions Division of Biotechne. Dr. Heger, you may now begin your presentation.
Thank you for that introduction. My name is Chris Heger, Director of Application Science for the Analytical Solutions Division of Biotechne. I'm here today to talk about socially distanced experiments and the importance of single lab accessible automation in driving high-quality proteomics research. In our lab, we have employed the following strategies over the past year to keep people safe. We started with the reduced workforce on-site to enable social distancing with remote work and staggered on-site hours. We washed our hands a lot. We wore and continue to wear masks in accordance with local, state, and federal guidance. And lastly, we were able to minimize the actual time spent in the lab and moved data retrieval and analysis to the home office. We're all familiar with the first three strategies here, but it's the last one that I want to dive into more today. Biotechne is a diverse, supportive organization with several brands to support your research needs, including ACD, Novus, Tocris, R&D Systems, and as the focus will be today, ProteinSimple. Together, these brands provide a broad portfolio of tools for research, including cytokines, growth factors, and small molecules, media, and supplements. Most of these are offered at GMP grade as well as research grade. We also have world-class immunoassays and ISH technology, and a wide variety of proteins and antibodies. In addition, ProteinSimple provides analytical instrumentation. Our goal is to offer all of these innovative solutions with flexibility and customization to fit your needs with the consistency, reproducibility, and scalability you require. Today I'm here to discuss one of our ProteinSimple platforms, Simple Western, in more detail. Analytical tools are constantly evolving to adapt to the rapidly changing research and development landscape. Sometimes these changes are geared towards allowing the researcher to make faster insights, as is generally the case with using today's smartphone compared to a desktop computer from 20 years ago. Sometimes these changes are meant to help provide a better view, as been the case for the microscope as it's evolved. When I think about ProteinSimple and Simple Western in the context of these pictures, I'm pretty confident placing this logo alongside these images. The traditional Western blot has been around for 40 years, and while being a key research tool, still persists in a form that looks pretty similar to what was available 40 years ago. There have certainly been innovations to help improve the overall process, but it is still just that, a process. It's a process that requires a lot of gear and a lot of time, up to 48 hours in some instances, and yields results that really aren't worth the effort. The Simple Western platform, on the other hand, takes the entire process, innovates, and automates it, all in one box, completed start to finish in as little as 3 hours. Having personally run thousands of blots the old way, I truly wished this platform had been around when I was running traditional Westerns. The Simple Western platform is the new gold standard in Western blotting. It's our completely automated capillary-based Western blot platform that removes the laborious, notorious Western blot from your workflow, giving you back time and confidence in your data with high precision and reproducibility. The Simple Western platform features fast time to results. The platforms offer fully automated size and charge immunoassays capable of completing a run in as little as 3 hours. The Simple Western platform offers quantitative data, either absolute or relative protein quantitation, with our fantastic compass for Simple Western software, which we'll talk about more today. The Simple Western platform has high throughput capabilities as well as flexibility. Up to 96 samples can be run in a single run on our high throughput instruments. We have a wide variety of multiplexing capabilities across the platforms, and several different platforms to choose from to best fit your needs. All of our Simple Western platform instruments offer outstanding reproducibility with low inter and intra-assay CVs. And lastly, really important for those folks working with precious samples, is low sample volume. The Simple Western platform uses less than 3 microliters per well and can get exquisite sensitivity of picogram level. The Simple Western immunoassay takes place in a capillary. Your sample, primary and secondary antibodies, as well as the separation and running buffers, are loaded automatically from a specifically designed plate. Jess starts by aspirating the separation matrix. This is followed by aspiration of the stacking matrix into each capillary. Next, your protein sample is loaded. And then the capillaries are lowered to make contact with running buffer. Voltage is applied to separate proteins by their apparent molecular weight. Once separation is complete, UV light immobilizes the proteins to the capillary wall. And with the proteins immobilized and separation materials cleared from the capillaries, Jess starts the immunoprobing process. Samples are first incubated with primary antibody, and then, in the case of chemiluminescence, an HRP conjugated secondary antibody. And finally, the chemiluminescent substrate. The chemiluminescent substrate reacts and is recorded by a CCD camera in a series of images over time. Now that I've introduced the technology a little, it's time to introduce the Simple Western family members. We have 3 small footprint instruments, Wes, Jess, and Abby, that can run Simple Western size assays in as little as 3 hours. They all have ultrasensitive chemiluminescence, and Jess has dual color fluorescence as well. And you can replex on both Jess and Abby. We also have 3 larger throughput instruments, Sally Sue, Peggy Sue, and the NanoPro 1000. These instruments can process up to 96 capillaries per run and can run size, IEF, or both depending on the model. All 3 of these larger instruments have ultrasensitive chemiluminescence detection as well. Since Simple Western is an automated platform, the reproducibility is outstanding. Whether you're looking within a run, like this PI3 kinase CDK6 multiplex, where we have CVs for 24 replicates under 10% for each target, or day-to-day and user-to-user, like here on the right, where we have 4 different users running experiments on 4 different days for one pretty good week of data, with CVs under 12% per day, and overall under 10% for 4 days with 44 replicates. Simple Western has multiple detection capabilities. The technology was founded with ultrasensitive chemiluminescence at its core. Here's a great example where we can detect as little as 0.4 nanograms per mil of CD19, a CAR T therapy target. But Simple Western has also evolved over the years and now includes 2-color fluorescence as well. Here's a beautiful example looking at EGFR signaling. Upon stimulation with EGF, the EGF receptor switches on by virtue of dimerization and phosphorylation. And we can monitor that with a phosphospecific EGFR antibody. In A431 cells, in the absence of stimulation, the EGFR receptor is not phosphorylated, as is shown in lane 1. But there is plenty of EGFR in these cells, as illustrated in lane 2, with a total EGFR antibody. However, upon stimulation with EGF, in lane 4, we see a large increase in the phospho-EGFR detected, as well as in lane 6, which is a multiplex of total and phospho-EGFR. Both ultrasensitive chemiluminescence and our dual color fluorescence are compatible with our reflex feature. Let's quickly define reflex. Reflex is defined as 2 rounds of immunoprobe and detection in a single capillary. This assay leverages the unique covalent immobilization chemistry at the heart of the Simple Western technology and is compatible with the chemiluminescence, fluorescence, and total protein assays. Importantly, since you're getting a second data point from the same capillary and same sample, using reflex reduces the cost per data point too. So in our normal workflow where we first separate a sample and immobilize it, the reflex takes place after the first detection. And an example output will be shown here where the 2 different lanes are for the actual different probes that happen in the same exact capillary. So in probe 1 for this capillary, we looked at a multiplex of HSP60 and GAPDH. And then upon replexing, the same exact capillary was interrogated for a multiplex of 1433 and PARC7. Let's take a quick look at a reflex example. Here's an example of how we leveraged reflex for breast cancer research, a field that is near and dear to me. Here we are looking at AKT signaling in MCF7 breast cancer cells in response to stimulation with IGF-1. For this experiment, we used a pan-AKT antibody detected with NIR secondary, shown in red here, and 2 different phospho-AKT antibodies to either phosphoserine 473 or phosphothreonine 308, detected with HRP secondary for chemiluminescence detection, shown in black. Upon stimulation with IGF-1, significant increases in AKT phosphorylation at both phosphosites is observed. In this assay, we use the reflex to go back over the same capillaries and interrogate them for the total protein loaded in probe 2, shown on the bottom in blue for these samples. Using the data obtained in reflex, we can correct our immunoassay data with that total protein information, which is automatically done by the Compass software. And that's shown by the area column, which is before correction, and the corrected area, which takes into account the total protein data. I wanted to spend a few minutes talking about Peggy Sue and what she brings to the table for size separations. Our higher throughput instruments, including Peggy Sue, shown here, can really help you get the most out of a precious sample. Let's walk through a quick example so I can show you what I mean. Let's imagine that these are 8 sample wells. Let's put 2 patient samples, sample 1 and sample 2, in those wells, where we are looking at whole cell and nuclear extracts of either basal or TNF-alpha treated cells. Placing only 5 microliters of sample per well, those samples will be interrogated for 8 different targets in this run for the NF-kappa B pathway. So whereas 5 microliters might get you a single well on a traditional Western for this assay, here we're getting you 8 times more data. The instrument is able to process capillaries in groups that we call cycles, and when this run is completed, Peggy Sue has elucidated an entire pathway, the NF-kappa B pathway in this example, and a loading control for these samples. So if you look down an entire column, These are all individual data points from unique capillaries on a PEGI-SU for individual targets. So a lot of data coming out of a single run on a PEGI-SU from single wells of sample. In addition to the size separation capabilities I've described, we also offer charge separation of proteins on PEGI-SU and the NanoPro 1000. In this separation mode, we use the same capillary capture and in-capillary immunoassay approach, but leverage the isoelectric point of the target protein. This allows one to look at subtle changes in a protein that cannot be detected by size separation, like, for example, phosphorylation, which only adds 80 Daltons to the protein. With charge separation, each phosphorylation acidifies the protein, moving it to the left of this PI scale. Let's look at an example of the MAP kinase proteins ERK1 and ERK2. These proteins require dual phosphorylation to become active kinases. Using a single antibody that detects both ERK1 and ERK2, when a helolysate is separated by charge, we see two main peaks, one for each ERK protein. When we look at HeLa cells that have been stimulated with EGF, we see a rapid increase in two acidic species at PIs 5.2 and 5.8, which are the diphospho species of ERK1 and ERK2. And we also see concordant decrease in the non-phospho forms. Additionally, there's a small amount of change in the monophospho ERK1 species. These changes can be quantified, and after EGF treatment, the two ERK proteins are nearly 90% phosphorylated. The same assay approach is used by researchers to look at lung cancer and emphysema. In the top panel are ERK signatures from normal lung tissue, shown in blue, and from lung cancer tumors, shown in red. There's essentially no visible difference between them, and we see predominantly ERK1 and ERK2. However, in the bottom panel, where normal lung tissue is compared to lung tissue from emphysema patients, there's a stark difference, and we see that this assay might be useful to help better understand this disease. These observations are quantifiable, and the data were shown to be statistically significant. The charge assay has been used in many other ways to look at other post-translational modifications, or in instances where proteins cannot be separated by their size. Simple Western has been supporting researchers for nearly 10 years, and today I can happily show you that the Simple Western has been used to help publish over 1,000 peer-reviewed publications, and is on quite an upward trajectory at this point. This table on the left does not include patents and doctoral thesis work either, so the number is actually a little bit higher than what I'm showing here. These publications are featured in diverse research areas as shown on the right. Everything from cell biology to clinical research, vaccine development, and bioprocessing. And these publications are in top-notch journals, with Simple Western data gracing the pages of Science, Cell, and Nature journals. Let's now talk a little bit more about how Simple Western can help maximize your socially distanced time, with a little bit more detail about its fast setup time, fast time to results, about how we have remote data access available, and a little bit more about our powerful all-in-one easy-to-use software. And I'm even going to provide some examples from the past year about how we at ProteinSimple have stayed successful while socially distancing using the Simple Western instruments. Simple Western assays on Wes, Jess, and Abby take as little as 30 minutes to set up. Importantly, to maximize social distancing, we have the run file saved to a network or share folder before we leave the lab or the site. Setting up the assay is easy, and that's partly because everything you need to run Simple Western is neatly organized and accounted for in our separation and detection modules. These high-quality reagents all undergo rigorous quality control to help ensure your data is the best it can be every time. We also offer a la carte options if you need a little bit more of something specific. I showed you what happens once the consumables are inside the instrument, but let's review the sample run prep workflow. First, you prepare your samples pretty much the same way as you would for traditional Western. You'll add a master mix to help denature the samples in the presence of SDS, often with reducing agent and a little bit of heat. And then the samples, along with primary and secondary antibodies, are aspirated and dispensed into the specific Simple Western plate shown here on the right. After everything's loaded into the plate and the capillaries get loaded into the instrument. And then you hit start and walk away. As we hit start in the Compass software, which controls the Simple Western instrument, we can set up remote data access for Simple Western as well. Last summer, we put together this remote tools protocol to help our Simple Western customers get their data while off-site. It's actually really straightforward. In the Compass Run Start menu, the software will ask you where you want to save the data. At this point, you need to point the save path to a shared folder that has network access. For more information on how to set up a networked folder, simply Google it. After hitting start, Wes, Jess, and Abby go to work and complete the run in as little as 3 hours. With only 30 minutes of hands-on time, these assays are the best and most efficient Westerns available. So we go home, and after 3 hours, the data is written to the network folder, which we now have access to remotely. Compass software is free and available for personal computer use as well. And the same software is used to design assays, monitor the instrument, and analyze data. So after you have your data in hand, you can analyze it, as well as plan your next experiment before returning to site. And while you're waiting for your run to complete, we have an additional feature for you. That's right, you can keep an eye on your run while you're away with Twitter. Let's take a quick look at how to set that up. Okay, inside the Compass software preferences, there's a tab for Twitter. After clicking on that section, we can now see various options. The first thing to do is allow Compass access to Twitter. You'll be asked to add a username and a password, and then to authenticate the login. Afterwards, we'll head back to the Compass preferences and send a test tweet to make sure your instrument is sending out messages. After confirming the test tweet, your instrument is ready to tell you how it's doing and when it's done. Let's take a quick check-in on an instrument's Twitter page. Here's a screen grab from a Jess instrument's Twitter page. This Jess is just getting started, so there's only a few test tweets, but this is the interface. You can get tweets from your instrument when the run is started or completed, or get a tweet if any errors pop up during the run, although that's a pretty rare tweet to get. We've accomplished a lot this year with these strategies and our instruments. In March 2020, our development team mobilized to develop a serology module for SARS-CoV-2. This module features 5 recombinant SARS-CoV-2 antigens that can be used to probe for individual reactivities from serum antibodies to various parts of the virus, thereby affording a deeper understanding of the infection. To test the reactivity of serum samples against these antigens, we analyzed serum samples that were confirmed positive for COVID-19 by PCR. In this analysis, serum samples are used as the primary antibody, and the secondary anti-human IgG-HRP conjugate supplied in the module was used for chemiluminescence detection. 2 PCR negative serum samples were included as negative controls. This analysis showed a high degree of reactivity of the PCR positive serum samples with the antigens included in the kit. As expected, there was little to no reactivity occurred with the PCR negative serum samples. When the relative peak areas were quantified, this analysis revealed a high degree of variability in IgG reactivity with each of the 5 antigens among the human serum samples. For example, PCR positive sample 4 had high reactivity with the nucleocapsid protein, while PCR samples 1, 2, and 3 had significantly lower reactivity with the nucleocapsid, but had increased reactivity with the spike protein. This is critical information, as it was shown that detection of the nucleocapsid is sensitive to early infection, while antibodies that target epitopes on the spike protein may be linked to neutralization. Overall, these data provided a much deeper characterization of the humoral response to COVID-19 than is possible with single antigen ELISA kits. We also were able to develop several other assays and applications in diverse research areas, including SARS-CoV-2 biology, where we looked at ACE2 and TMPRSS2, in PROTAC degradation of neosubstrates, and lastly, in the impurity analysis of AAVs. Here's a closer look at one of those application notes where we leveraged Reflex to monitor impurity analysis in AAV process samples. AAVs are a promising gene therapy delivery tool, and as such are high focus among biotherapeutic manufacturers. We used an anti-AAV VP1, VP2, VP3 antibody in chemiluminescence detection, followed by Reflex with total protein on these AAV in-process samples for serotypes 1, 4, 7, and 9. The assay only required 50,000 copies of AAV sample per well, which is great considering how challenging it can be to get enough AAV samples to work with. And as expected, the antibody reacted quite well with AAV 7 and 9, but not with AAV serotypes 1 or 4. The data can easily be quantified for both viral proteins shown on the top or the total protein shown on the bottom. And as I've described previously, using the total protein, we can normalize for the viral proteins within these samples and account for any differences in the loading. So I've mentioned that we've done quite a bit of work in the last year, and we did a lot of it remotely. And part of that work is making figures. We rely on our Compass for Simple Western software a lot. And today I wanted to show you a quick example of how the newest version, Compass 6.0, is ideal for making high-quality, publication-ready figures. For this discussion, we're going to use a multiplexed assay that I actually described a little bit earlier, but here's the entire assay. So this is a Jess run looking again at those MCF7 cells in the absence or presence of stimulation with IGF-1. And we're probing with different multiplexes for AKT signaling, either phospho antibodies with total pan-AKT for either specific phosphosites for the isoforms or for non-phospho AKT1 and AKT2. Okay, so there's 6 different multiplexes here, and all of these data are going to be replexed for total protein. Let's take a look at how the run shakes out. From this one run, there are actually 3 complete sets of data. Because it's a multiplex in the immunoassay, we have pan-AKT detected at NIR, shown in the top picture, and in our chemiluminescence detection, we use either phospho-AKT or AKT isoform-specific antibodies. And again, in the reflex, we have the total protein, which is shown on the right. So it's a lot of data. Let's see how we can make that into a nice figure. With Compass 6.0, making the publication quality figures inside of Compass is really easy. We have a new lane annotation feature shown here on the left. Upon selecting the Annotations tab, the software is going to ask you to generate a crop region. After generating a crop region, shown on the right, the software allows you a number of different options, including lane labels, band labels, figure titles, figure notes, and other settings. And taking those same data that we started off with, I've made 3 different crop regions, one for each of the detection channels, but for the same capillaries. Shown here is the result of that image analysis on the right. So I've added a title indicating what the experiment was about, as well as changed the angle of my labels and annotated the individual peak names as well. I didn't have time to show you how I did all of that today. We have a lot of great help in our Compass for Simple Western User's Guide. The Biotechnic Academy is our online training center that provides all of the training support needed to get started. After self-installation of a WES, JES, or ABBY, simply start your designated instrument learning plan. Shown here is an example of our JES instrument learning plan. The Academy not only teaches you, but it also connects you with your local field application scientist. Rest assured that every user at your site can visit the Academy at any time from anywhere. Not only does the Biotechnic Academy provide a breadth of training content to get you up and running on a variety of instruments and platforms, but it also serves as a great resource to continue to get a great scientific content to keep our customers growing as scientists as well. In summary, Simple Western maximizes your social distancing strategies by virtue of its fast prep time and fast time to results, and the ability to set up remote run data access. Since most Simple Western instruments are plug and play, once you have yours delivered, you can install it when you're ready, essentially just by plugging it in. And once you're ready to play, we've got fantastic Biotechnic Academy for online training. And lastly, as I showed today, our newest version of Compass, Compass 6.0, supercharges data analysis with powerful figure annotations that can be made anywhere, anytime, without being connected to any instrument. For more information, visit us at ProteinSimple.com. And with that, I'm happy to take some questions. Thank you.
Thank you, Dr. Heger, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question, are there any limitations in reviewing the data remotely?
Yeah, hi, and thanks for the question. And as I mentioned, our Compass software is available is free for anybody to download and use on a personal computer. So, one of the main limitations with some software packages is you got to have a specific license for it, and we definitely don't have that in front of you. I could imagine some related to, firewalls on sites where you work, that might be of some challenge, and I would recommend you engage your IT department as well as reach out to our tech support to help you.
Great, thank you, Dr. Heger. Our next question is, what is the cost for consumables, or does the Biotechnic Academy cost money?
So the Biotechnic Academy, which I described at the end there, that is a free academy as well. So any user can sign up. So if you have 5-10 people in your lab, they're all able to get into the academy. In terms of consumables, I like to tell folks that I'm a scientist and I don't have all those numbers at the top of my mind. What I do like to remember is the cost is comparable to that of a traditional Western. If you boil it down to the nuts and bolts of it, the main difference is how much hands-on time, the FTE time of a traditional Western. That's a lot of expense that you don't run into with Simple Western.
Great. Thank you, Dr. Heger. And our next question is, what is the volume inside the capillary?
Yeah, these are pretty small capillaries. I didn't describe that. They're 5 centimeters long, 100 micron internal diameter, which works out to roughly 400 nanoliters of total internal volume in the capillary. And for the size assay that I described, we load a small sample plug, which is approximately 40 to 50 nanoliters.
All right, great. We have another question coming in. You showed some nice data for Reflex, but mainly showed using total protein in the second probe. Can I use Reflex with a second immunoassay?
Yeah, that's a good question. There are a number of different approaches or assay combos you can use with Reflex, and those include, obviously, as I showed a lot of today, the immunoassay followed by a Reflex of total protein. But you can do 2 sequential immunoassays. Those could be combinations of our NIR and IR and Chemi, or any combination therein.
Great. Thanks again, Dr. Heger. Our next question is, which instruments are compatible with Reflex?
Yeah, so the Reflex feature was launched, I believe, a little bit over a year and a half ago, and it's available on our Jest instrument as well as our newest Simple Western family member, Abby.
All right, perfect. Thank you. Next question. Is the SARS-CoV-2 module available for all Simple Western instruments?
Absolutely. The core components of that kit work with the little boxes, as I like to call them, Wes, Jess, and Abby, as well as our higher throughput size-based separation instruments, either the Sally Sue or the Peggy Sue.
Great. I love those names, by the way. Next question. Have you compared the SARS-CoV-2 module to an ELISA?
Yeah, that's a question that comes up quite a bit. ELISA is obviously a gold standard, especially in serology assays. And we did do that comparison, and it turned out really well. We compared the Kintaro ELISA, which is a Biotechnic collaborative effort, and we produced that kit, and we ran a series of samples and ran a comparative analysis after the fact with Simple Western, and found a really high percentage of agreement for positives and an extremely high positive agreement for the negatives as well, demonstrating that the Simple Western assay sees essentially the same thing as the ELISA, but also provides the additional information of those other SARS-CoV-2 antigens.
All right, great. Thank you again, Dr. Heger. Our next question is, you showed some nice AAV work. Which antibody did you use for the VP detection?
Yeah, thanks. The AAV work is something we're quite proud of. What I showed today is from our most recent application note, which I encourage people to go check out. The antibody we're currently using is from a company called ProGen. It's a VP123 antibody. It works pretty well, as I showed a nice example of today.
And it looks like we have time for one last question. Can I use Compass 6.0 on older data to make new figures?
Yeah, that was a big one that even folks within my own team were asking. The ability of Compass 6.0 to take out a bunch of the steps that you typically would do in PowerPoint is really powerful, and absolutely. So if it's an older WES or Jest file, pre-Compass 6.0, it'll certainly be usable in Compass 6.0 to make those fancy figures that I showed.
Well, thank you again, Dr. Heger, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Biotechne, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.